Hey everyone, I think this will be our last video in chapter 6. So now we're going to talk about how we isolate viruses and how we can grow viruses. Viruses can't be grown in a petri plate like bacteria are. They have to be grown in living cells. So viruses don't replicate outside of a cell. And because of this, we have to find different types of cells. So bacteria are used to grow bacteriophages, and in animals we often use chicken eggs to grow animal viruses. And so if you look over here, once we infect the cells and we allow the bacteria or the viruses to grow, we then can filter the virus from the other cells. And we can do this by using special filters. So here we have a filter that is 5 micrometers in size, but a filter that is 5 micrometers can actually allow bacteria and viruses through. So if we use a filter that is 2 micrometers or 200 nanometers in size, then only the viral particles can get through, and we then have the virus that we can use for whatever purposes we're going to use it for, maybe to make a vaccine, or maybe we're going to do some study on the virus. So here is a figure showing the utilization of animal embryos, so chicken eggs and turkey eggs, where we infect them and we use those cells or those living structures to grow our animal viruses. And then with that we can get the viruses out and we can produce our vaccines or do whatever studies we're doing. In the 1940s, the late 40s, there was a lady named Henrietta Lacks, and this is her right here. She has a cell line that is used today. It's known as the immortal cell line, and virologists use it today to study multiple viruses, including poliovirus, AIDS virus, human papillomavirus. And so, and actually my daughter does research using these HeLa cells, and she studies HPV and cervical cancer in her research. So what HeLa cells are, these are cells that came from Henrietta Lacks' cervix. She had cervical cancer and she died at a young age because of this. What is kind of interesting, or kind of a crazy story, is that her cells were taken from her to, do testing. Her cells were sent off to laboratory for testing, and they grew really well. And so these lab kept the cells and grew the cells, producing new cells. And her family was never told. She was never told. She ended up dying. Her family never received anything from these cells, and the cells themselves are used continuously in research. So it's kind of like, shouldn't the family have received something for this? I mean, it was Henrietta's cells that are allowing all this research to be done, but people didn't even give her cells credit or give the family credit. So I at least like to give a little credit to this individual who, unbeknownst to her, donated her cells. I don't think it was really a donation, but and her cells are used, and we have been able to find cures, vaccines that can keep polio, well, to cure polio, or not cure polio, but to keep people from getting polio. And we use them in studying things like cervical cancer, other forms of cancer, and other research. So now let's jump into viroids. Viroids were discovered in 1971 by Theodore Diner. These are short strand circular RNA structures. They only have RNA. They have no protein coat. They are obligate intracellular pathogens to plants only. There are no viroids that actually infect humans or animals, I should say. And then we have virions, or I'm sorry, not virions, virusoids. Virusoids are single-stranded RNA structures that only work if there's a helper virus available. They typically infect plant cells, but they can infect human cells. And so an example of a virusoid is a virusoid that infects, so the hepatitis delta virusoid infects humans, but has to have a helper virus, and the helper virus is hepatitis B. So if you have hepatitis B, you then can get the hepatitis delta virusoid as well. And that causes a huge problem. So they can't infect cells alone. They can't do anything alone. They have to have the help of a helper virus. And then we have prions. So Stanley Prusiner is the individual who identified the first prions in 1982. He was studying scrapie, which is a prion disease in sheep, similar to the prion disease known as Creutzfeldt-Jakob in humans, and mad cow disease in animals, or in, not animals, in cows. A prion is a proteinaceous infectious particle. That's where the name comes from. So it's basically a protein that misfolds and causes other proteins to misfold. This occurs in the nervous system, and it causes all kinds of damage to the nervous system. So some of the first areas where you see prions, or how prions can be, I won't say areas where you see, I'll say prions can be received via blood transfusion, as well as if you eat protein or eat food infected by prions. So like in certain countries where eating the brain of a human is common, prion diseases are going to be more common. So Kuru is a disease associated with cannibalism. And in certain countries where Kuru was more common, they had a certain ritual. When family members died, they would eat their family members. And so they would cook the family member, but since prions don't break down like other proteins, they would then eat the meat. And so women and children ate after the males. So the males would eat first, they would get the good food, and then the women and children would eat, and they would get the nervous tissue and other material. And so you would see this prion disease, Kuru, spread in these villages where cannibalism was more common. Kind of creepy, right? So this is what a prion does to the brain. Here's a normal brain. Here is a brain that has a prion. This is Creutzfeldt-Jakob disease, which is the human prion, or the prion that attacks the human brain. This can be caused by a genetic mutation, but oftentimes it occurs because of taking in protein from an organism that has the disease. So eating the cow meat from a mad cow disease cow, or eating the brain of someone with kuru. So this causes transmissible spongiform encephalopathies in humans. So this is normal brain tissue. This is what brain tissue looks like with a patient with Creutzfeldt-Jakob disease. This is a neurodegenerative disease. It causes, there's no cure. There's only symptomatic treatments, so you're treating the symptoms. Eventually, individuals will lose, so they will be depressed, they will lose their ability to walk, they'll lose their ability to go to the bathroom on their own, they'll be unable to swallow, they'll have muscle spasms. Eventually, their brains will be broken down to the point where they will have dementia and become bedridden completely. They may not even speak, and it always ends in death. So this is a very serious disorder, and prions are serious, non-living, proteinaceous materials. And the worst part about prions is they're very hard to get rid of. So you can put a prion in an autoclave and run it for 4 hours, and it still will be able to infect an individual. So that's kind of crazy, right? This was a little longer of a video, 12 minutes, apologies, but I am done. So I'm going to stop here, and I'll upload these videos. You guys have a great week. Bye.